Hello, dear pharma aspirants. Welcome to GDC. So basically, we have started a Tejas series, which is basically a question answer series for railway pharmacist exam. So in the today's video, we basically talk about biochemistry subject. The topic which is covered today is your carbohydrate and its metabolism as well as organ function states. Let's come to the first question. So first question is a general formula of monosaccharide. This is a very very important question. What is the general formula of monosaccharide? Your options are option A CnH2ON, option B C6H12O5N, option C C6H10O6N, yeah, option D C6H10O6N. So what is the general formula of monosaccharide? So here the right answer is option number A, CnH2ON, or sometimes it is written as CnH2nON. This is the general formula for monosaccharide. So this is there are single unit of what is carbohydrates? So carbohydrates are basically polyhydroxyaldehyde or ketone. So what are they? They are polyhydroxyaldehyde ketone. Polyhydroxy means the aldehyde or ketone having more number of hydroxyl group, or we can say OH group. Okay, these sugar are the simplest non-hydrolyzable. Monosaccharides are basically simplest and non-hydrolyzable. Your example is glucose and fructose. Glucose is a principal carbohydrate that is basically found in your human circulation. Okay, next come to the next question. Pentose sugar. So what is pentose sugar? Which of the following, the question is, which of the following is an example of pentose sugar? So options are dihydroxyacetone, ribulose, erythrose, your glucose. So here the right answer is ribulose. Okay, so the when n equal to 3, example is your dihydroxyacetone. Okay, so here, yes, triose. When n equal to 3, we call it as a triose. Example is dihydroxyacetone and glyceraldehyde. When n equal to 4, we call it as a tetrose. The example is your erythrose and erythrulose. When n equal to 5, here n equal to 3, here n equal to 4. When n equal to 5, it is called as pentose. Example is your ribose, ribulose, deoxyribose, xylulose. These are comes under your basically your n equal to 5 or pentose sugar. Next is hexose sugar when n equal to 6. So example is your glucose, galactose, mannose and fructose. These are comes under hexose class. Next is heptose where n equal to 7. The example is pseudoheptulose. Okay, these triose are again of two type, uh, aldose and ketose. So what is aldose? Aldose having aldehyde functional group, ketose having ketone functional group. Okay, next, a heteropolysaccharide among the following. Which of the following is a heteropolysaccharide? Okay, inulin, cellulose, heparin, or dextrin. So question is, which of the following is a heteropolysaccharide? So heteropolysaccharide, what is, there are two polysaccharides. So polysaccharides, if we'll talk about polysaccharides, so these polysaccharides are broadly divided into two class. One is your homopolysaccharide, another one is your heteropolysaccharide. So what is homopolysaccharide? When similar type of a uh, compound are there to form a polysaccharide is called as homo. When different type of compound is there, we call it as a hetero. So here the right answer is heparin. So what is the polysaccharide? Polysaccharides are the polymer of monosaccharide. When there is a similar type of polymer is there, we call it as a homopolysaccharide. Whereas when the different type of polymer is there, we call it a heteropolysaccharide. So the example you have to remember. Okay. So the example you have to remember, homopolysaccharide example is starch, dextrin, inulin, glycogen, cellulose. Heteropolysaccharide example is your hyaluronic acid, chondroitin, and heparin. Okay, let's move to the next question. The next question is telling that alpha D glucose and beta D glucose are what? So this question is very, very important from exam point of view. What is alpha D glucose and what is beta D glucose? So alpha D glucose and beta D either their options are either they are stereoisomer or epimer or anomer or keto-aldo pair. Here the right answer is option number C, anomers. So anomers are what? If two monosaccharides, if two monosaccharides differs from each other, differs from each other in a single carbon, we called it as a anomer. Okay, so if the alpha and beta form, the alpha and beta form of D glucose are known as anomer. They are different from each other in a configuration around a single carbon, that is C1 anomeric carbon. Similarly, alpha D example is alpha D glucose and beta D glucose are comes under are example of anomer. Similarly, alpha D fructose and beta D fructose are example of anomers. Okay, next question is two sugar which differ from one another in a configuration around a single carbon. Okay, so if so alpha D glucose and beta D glucose are anomers. Yes. Next question is two sugar which differ from each another. See, when two sugar moiety, two sugar moiety are differ from each other in a single carbon atom, we called it as a apimer. Okay, what we called it as a apimers. Okay, question is what the question is asking basically. The question is asking that when two sugar differ from each other and each one another only in a configuration around a single carbon, they are differ from each other in a single carbon. In one particular carbon, thus all the structure will be same. In only one carbon, the structure is differ from each other. Then we call it as a epimer. Okay. So when two monosaccharide differ from each other in the configuration around a single asymmetric carbon. Okay. Asymmetric carbon means a carbon having attached to it four different functional groups. For suppose 4H, H, CS3, or we can write it as a CH2 CH3. So we call it as a what we can call it as a anomeric carbon or asymmetric carbon. We call it as a asymmetric carbon. When glucose and mannose are the C2 epimers, whereas glucose and galactose are C4 epimers. Glucose and mannose are C2 epimers, whereas glucose and galactose are C4 epimers. Okay, next move to the next question. Sucrose consists of. Okay, sucrose is a if sucrose is a so question is sucrose is basically consist of what? Glucose or glucose fructose, glucose galactose, yeah, glucose mannose. So you have to remember that what are the constituent of a sucrose. So sucrose is basically consist of GF. Remember sucrose as a GF, glucose and fructose. So sucrose is also known as cane sugar, mostly produced. This is basically produced in sugar cane as well as beet. Okay, sucrose is made up of alpha D glucose plus beta D fructose. And these two monosaccharides are held together by alpha 1 and beta 2 between C1 of alpha glucose and C2 of beta fructose. This statement is very, very important. So you have to remember these two monosaccharides are held together by what bond and in which carbon. So these two monosaccharides are held together by glycosidic bond, that is alpha 1 beta 2. The glycoside bond is alpha 1 and beta 2. Okay, and which carbon? Alpha C1 of glucose and C2 of fructose. Okay, sucrose is the major carbohydrate produced during photosynthesis. Okay, sucrose is the most abundant among the naturally occurring sugar. This is the most abundant naturally occurring sugars. Okay, next moves to the next question. So next question is, which of the following is a disaccharide? Okay, so what is disaccharide? This is consisting of two sugar moiety. Example is sucrose, fructose, glucose, galactose. Just now we have discussed sucrose. Sucrose is consisting of alpha. Sucrose is consisting of alpha D glucose plus beta D fructose. Okay, when these two combine, we basically get what? We basically got sucrose. If the position we ask alpha 1 and beta 2. Carbon if ask C1 of glucose and C2 of fructose. C1 of glucose and C2 of fructose. Okay, next move to the next question. Yes, we will see the explanation. Disaccharide means these sugar are hydrolyzed into two molecules of monosaccharide. When does this sugar hydrolyze? We will get two monosaccharide. Example is your sucrose, your lactose and maltose. These three forms under disaccharide. Trisaccharide example is these sugars are hydrolyzed. These sugars are hydrolyzed into three molecule of monosaccharide. Example is raffinose and ketose. Next is tetrasaccharide. These sugars are hydrolyzed to four molecule of monosaccharide. Example is lincose and maltotetrose. So you have to remember the example which sugar is comes under which class of oligosaccharide. Okay. Next is the breakdown of glucose to pyruvic acid is called as what? So what is the question is the conversion of glucose to pyruvic acid. So this process is basically called as oxidation of glucose, and the method is basically called as glycolysis. So what is glycolysis? So glycolysis is a process by which glucose converted to pyruvic acid or another compound can be formed, that is lactic acid. So this is basically depending upon the oxygen. If oxygen is present, then pyruvic acid is formed. If oxygen is absent, then lactic acid is formed. What is glucogenesis? So glucogenesis means synthesis of glucose from glycogen. We call it as a glucogenesis. Glycogenolysis, that is breakdown of glycogen. Okay. So in TCA cycle, we basically there is a release of energy. Okay. So TCA cycle glycolysis, this is the oxidation of glycolysis, basically the process. This is also known as EMA pathway, MDEN, Mayer-Hoff pathway. This means the oxidation of glucose to pyruvate and lactate. Next is citric acid cycle or Krebs cycle or tricarboxylic acid cycle. The oxidation of acetyl-CoEnzyme to carbon dioxide. Okay. Krebs cycle is a final common oxidative pathway for the. This statement is important. Why important? This statement is saying that Krebs cycle is the final common oxidative pathway for the carbohydrates, fats, amino acid through acetyl coenzyme. Okay. Next question is gluconeogenesis is the synthesis of glucose from the non-carbohydrate. Gluconeogenesis synthesis of glucose from non-carbohydrate sources. Glycogenesis synthesis of glycogen from glucose. Glycogenolysis is breakdown of glycogen into glucose. Next, coming to the next question, the only significant enzymopathy Emden pathway is of carboxypeptidase, carboxypeptidase. Only significant enzymopathy of carboxypeptidase, elastase, aminopeptidase, या pyruvate. Your option, right option is pyruvate. So what is this pyruvate? Glycolysis means your oxidation of glucose into pyruvate and lactate. So this basically oxidation will lead to formation of pyruvate. So moving to the question number 10, which of the following is a rate limiting enzyme in glycolysis? This question is asked several times in exam. Which is a rate limiting enzyme? Hexokinase, phosphofructokinase, pyruvate carboxylase, pyruvate kinase. So rate limiting enzyme means this is the important enzyme, the slowest step, and the important enzyme which basically control the rate limiting step of this glycolysis. The answer is your phosphofructokinase. Okay, he will talk. So most of the reaction of glycolysis are reversible, and where it is takes place, this glycolysis always takes place in cell cytoplasm. Where it takes place, it takes place in cell cytoplasm. However, three steps are catalyzed by enzyme hexokinase or glucokinase, phosphofructokinase, and pyruvate kinase. These three are irreversible. Okay. Um, so how I remember? I remember as HP Pavilion. So a laptop HP there is a laptop of HP Pavilion series. I remember like that. H for hexokinase, P for phosphofructokinase, P for pyruvate kinase. These three are irreversible. These three are irreversible. These three stages are mainly regulate the glycolysis, the reversal of glycolysis with an alternate arrangement made through three irreversible leads that's basically called as gluconeogenesis. Phosphofructokinase is the most important regulatory enzyme in glycolysis. This is the rate limiting committed step. Okay. See, GDC has launched a All India Pharmacist crash course for Bihar DSSB pharmacist. Okay, this basically live classes are already started from the 8th August. Okay, so this course is now this course is in offer. Now the offer price is 2,999. So what you will get in this course? You will get live classes, recorded video lectures, online test series, PU PUQ of MCQs and E-notes. Okay. Now moving to the next questions. Which of the following conversion requires involvement of ATP? So question is from the which of the following conversion requires basically ATP for the catalyzation of this step? 2-phosphoglycerate to phosphoenol pyruvate. Question is requires. Here ATP is basically question is basically asking is in which step ATP is utilized. So options are 2-phosphoglycerate to phosphoenol pyruvate step, glucose 6-phosphate to fructose 6-phosphate, 3-phosphoglycerate to 2-phosphoglycerate, या fructose 6-phosphate to fructose 1,6-bisphosphate. Here the right option is fructose 6-phosphate to fructose 1,6-bisphosphate. So let me summarize the glycolysis for you. This basically all the four steps which the option contains are these are the steps of glycolysis. So let's summarize the glycolysis. First step is glucose convert to glucose 6-phosphate. Here ATP is utilized. Similarly, conversion of glucose 6-phosphate to fructose 6-phosphate. Now fructose 6-phosphate to fructose 1,6-phosphate. Here also ATP is utilized. So question was which of the following conversion requires ATP? Fructose 6-phosphate to fructose 1,6-phosphate. Okay. So here this step is there. So this is basically your correct answer. So if the question was asked is in which step basically the ATP is produced. So here the step 6 where NAD convert to NADH because 1 NADH2 is equal to 3 ATP. Here 1 ATP, here 1 ATP. ओके 3 + 1 यहाँ पे हो गया Okay, 4 ATP. Now, okay. So these are the way how the ATP, yes, another one ATP is produced here. One ATP. So this is the overall overview of glycolysis steps. Okay, so this question is very very important from exam point of view. All the next question is, if we talk to the next question, all enzymes involved in glycolysis. Yes, just now I have discussed. So all the enzymes involved in glycolysis are present in where? Mitochondria, blood, cytosol, cell wall. Okay, you have to remember one thing. So if we'll talk about glycolysis, if I will talk about glycolysis, so this glycolysis is basically carried out in cytosol because all the enzymes are present in cell cytosol. If we'll talk about TCA cycle, if I will talk about TCA cycle, all the reaction will be carried out in where? In mitochondria. In mitochondria. You have to remember like this. Okay, your correct answer is cytosol. So glycolysis is defined as a sequence of reaction converting glucose into the pyruvate or lactate with production of ATP. The glycolysis takes place in cell of the body. The enzyme of this pathway basically present in the cytosomal fraction of the cell. So this is basically present in the cytosomal fraction of the cells. Moving to the next question, the number of ATP produced when the molecule acetyl coenzyme A is oxidized through citric acid cycle. Okay, this is very, very important. Question is the how many ATP is produced when one molecule of when a acetyl coenzyme A oxidized, how many ATP is produced? So your right answer will be 24. Okay. Okay, to why 12? Because see, when the TCA cycle occurs, remember when TCA cycle, this is basically occurs two steps process. Okay, so in one step we get 12 ATP. So total, if after completion of total two steps, so total ATP we got 12 plus 12 is 24. As it is asking about 1, so your right option will be option number A, 12. See, the what are the steps where ATPs are produced? So conversion of isocitrate to oxaloacetate, 3 ATP because 1 NADH is formed. Okay, similarly 3 ATP means 1 NADH. 1 ATP, 2 ATP means FADH, 1 FADH. 3 ATP means 1 NADH. So total ATP is, number of ATP is 12 ATP. Okay. Next, move to the next case question. The total energy gained from one molecule of glucose under aerobic condition. So question is, during aerobic condition, the total number of ATP that will be obtained during the total aerobic condition means presence of oxygen. Okay. So your option are 8, 38, 12 or 36. So here the right answer will be option number B, 38 ATP. Remember, during aerobic condition, the complete metabolism of glucose will lead to 38 ATP. How 38? Remember, 24 will be from TCA cycle, 24 ATP from where? TCA cycle, and 8 ATP from glycolysis. So if we total add, it will be 38. Okay. See, net ATP in this step is glycolysis. This is your TCA cycle. So total ATP is 8 in the glycolysis. As I told you, from one reaction we get 12 ATP. From two reaction total we get how much ATP? 24 ATP. So when we add, the total net ATP will be 38. Next question is, what is the process of breakdown of glycogen to glucose? When glycogen is breaking down into glucose, this step is called as glycogenolysis, because when we talk about breakdown, so breakdown with the lysis is term basically stands for breakdown process. When the term genesis is there, if a term genesis is there, genesis means synthesis. Okay, so this term, the difference between two terms, you have to remember. Okay, next move to the next question. Yes, option C will be the right answer, glycogenolysis. If I'll just summarize, glycolysis is the oxidation of glucose to pyruvate and lactate. Gluconeogenesis is synthesis of glucose from non-carbohydrate sources such as amino acid and glycerol. Glycogenesis means synthesis of glycogen, and glycogenolysis means breakdown of glucose. So the rate limiting enzymes are phosphofructokinase in glycolysis, fructose 1,6 bisphosphate in gluconeogenesis. In glycogenesis, the rate limiting enzyme is glycogen synthetase, and in glycogenolysis, the rate limiting enzyme is phosphorylase. The rate limiting enzyme is phosphorylase. Okay, so moving to the next question. The next question is telling that which of the following test is commonly used for assessing kidney function. So the major test that used for the assessing kidney function is, our options are liver function test, lipid profile, renal function test, thyroid function test. So here the right answer will be renal function test. So renal function test is basically a test which is basically detect the function of kidney. Okay, so whether the kidney is working properly or not. Another test is there, which is basically measurement of GFR, that is glomerular glomerular filtration rate. Okay, GFR we can measure. So the agent which is used for the measurement is inulin. Inulin is basically used. Okay, moving to the next question. Which of the following test is commonly used to measure the cholesterol triglyceride level in the blood? Which test is used to measure the cholesterol triglyceride? Option is liver function test, lipid profile test, renal function test, and thyroid. Question is measurement of cholesterol and triglyceride. When we talk about cholesterol and triglyceride, we basically measured by your lipid profile. So the options are, sorry, your right answer is a lipid profile test. So lipid profile basically measure your lipid amount in blood. This lipid includes total cholesterols, lower density lipoprotein, cholesterols, high density lipoprotein cholesterol, and triglycerides. Okay. Next, what is SGOT stands for? So SGOT stands for, with your options are serum glutamate oxaloacetate transaminase, serum glutamic oxaloacetic transporter, serum glycine oxaloacetate transaminase, serum glucose oxaloacetate transporter. Here the SGOT stands for option number A, serum glutamate oxaloacetate transaminase. This full form is very important from exam point of view. One is SGOT, another is SGPT. Okay, these two terms you have to remember because directly the full form can be asked in a exam. Okay, SGOT is serum glutamate oxaloacetate. This is known as also known as aspartame transaminase, also called as AST. We call it as a AST. Okay, so this enzyme is that convert aspartate to alpha ketoglutarate into oxaloacetate. Another test is SGPT, that is serum glutamate pyruvate transaminase, also known as ALT. Also known as ALT. Okay, these two tests is very important in case of to check whether the any type of disease, any type of infection or inflammation occurs in liver or not. Okay, the functional unit of kidney is, what is the functional unit of kidney? Option is glomerulus, nephron, corpuscle, calyx. So option, your right answer is, which is the functional unit? So nephron is the functional unit of the kidney. Okay. So nephron is the functional unit of the kidney responsible for the filtering blood and forming urine. Okay, each nephron includes glomerulus, which is filter the blood, and a blood tubule where reabsorption secretion takes place. While glomerulus is a part of the nephron, it alone is not functional unit. Corpuscle and calyx are the other part of the kidney involved in the filtering and collection process. Do not but do not function independently as a nephrons do. Normal blood urea level. So, what is the normal blood urea level? So blood option is range is 5 to 20 mg per deciliter, 1 to 5 mg per deciliter, 2 to 7 mg deciliter, or 2 to 10 mg per deciliter. Your right option is 5 to 20 mg normal urea level in blood. Okay. Uh, blood analysis, the normal to broadly evaluate the kidney function, serum creatinine and blood urea level basically estimated. The impaired when the glomerular GFR is impaired, the blood urea nitrogen level basically increases. So that basically indicating the kidney the person is suffering from any type of kidney disease. Okay, for normal serum creatinine level in male is 0.74 to 1.35. In adult women, 0.59 to 1.04 mg per deciliter. Okay. Now, as the RRB pharmacist exam will be going to happen in upcoming months. So we have launched a new batch for this RRB pharmacist course, so which contains 200 live classes, 500 plus recorded video lecture, 400 plus online test series, 100 plus previous year MCQ. Along with that, you will get e-notes. Okay. You can attend the free pharmacist marathon in our YouTube channel. Okay. So this the you can join this batch. The price now the offer is going on. Now the price is 2,999. Okay. So moving to the next question, which reagent is used for analysis of urobilinogen in urine? Okay, which reagent is used? 𝘿𝙬𝙎𝙬 reagent, Vanderburg reagent, Faucet's reagent, Tollens reagent. Here the right answer is 𝘿𝙬𝙎𝙬 reagent. So remember, urobilinogen in urine. Among the amount of urobilinogen present in the urine basically depend upon the amount of bilirubin enter into the intestine. Urobilinogen is estimated semi-quantitatively by which reagent? 𝘿𝙬𝙎𝙬 reagent. 𝘿𝙬𝙎𝙬 reagent prepared by dissolving 4 gram of para-dimethylaminobenzylaldehyde in 100 ml of concentrated HCl, then 100 ml of distilled water. Okay, when this reaction as we are dissolving in HCl, this reaction is carried out in acidic medium, the formation of a pink to red color. Significance is an increased urobilinogen in urine indicates the hemolytic jaundice. What it basically indicate? Hemolytic jaundice. Moving to the next question, which following test is used for the early identification of renal impairment? Which of the following test is basically used for the identification of renal impairment? Your options are creatinine clearance, urea clearance test, acid load test, urine concentration test. So your right answer is creatinine clearance, because creatinine, urea and creatinine clearance is the first and most important biomarker for the estimation of renal impairment. Okay, the kidney function test basically consisting of urine analysis, tubular function test, and GF test, glomerular filtration test. So tubular function test including urine concentration test and acid load test. GFR test includes clearance test, that is creatinine clearance test, urea clearance test. And blood analysis includes serum creatinine analysis and blood urea analysis. Creatinine clearance is the method for the measurement of amount of creatinine excreted in your through your urine. Creatinine clearance basically defined as the volume of plasma that would completely clear the creatinine per minute. Okay. Next is creatinine. Creatinine is basically excreted in a constant and not affected by the body metabolism factor, but due to this several other disease condition, this clearance level basically reduced. Okay, so GFR is basically measurement of glomerular filtration rate. This is basically measured with the help of a carbohydrate, that is inulin. Okay, so normal range is around 120 to 125 ml per minute. So if a lower GFR is sensitive indicated by creatinine clearance less than 75%. Okay, moving to the next question. Aclouric is a type of toxic jaundice, hemolytic jaundice, hemopic dysentery, obstetric jaundice. So this is a type of hemolytic jaundice. So achloric jaundice is without bilirubinemia associated with elevated unconjugated bilirubin. So this is basically associated with unconjugated bilirubin. Okay, that is not excreted by our kidney. Okay, so moving to the next point, familial achloric jaundice is another name for the hereditary form of another name for the hereditary pre-hepatic jaundice. Okay, so this yeah, unless the patient is the major symptoms is unless the patient concurrently affected by the hepatic dysfunction or is experiencing hepatocellular damage, that is a damage to the liver cell, and the liver does not contribute to the to this type of jaundice. Okay, an indication of renal function which is used for the estimation of glucose glomerular filtration is serum urea, serum alkaline phosphatase, blood urea nitrogen, or serum creatinine. Your right answer is serum creatinine. So serum creatinine is the one of the parameter for the detection of glomerular filtration rate. Okay, glomerular filtration rate is a critical indicator for kidney function. Okay, serum urea, blood urea nitrogen, we call it as a burn level, provides some information about kidney functions. Okay, serum alkaline phosphatase is an enzyme that related to liver, bone, and other tissue, is not specifically used for the estimate of GFR. Serum creatinine is also most commonly used and reliable marker for GFR. This is very, very important. You have to remember. Serum creatinine is most commonly used and one of the reliable marker. So moving to the next question, which organ does jaundice affect? So jaundice is basically affect which organ? Lungs, heart, liver, stomach. So your right answer is your liver. So liver is the prime organ which is susceptible for the damage during jaundice condition. Jaundice is a yellow color into the skin, the mucous membrane, and the eyes. The yellow color is basically due to what? The bilirubin, which is a byproduct of iron metabolism. Jaundice is often significantly problem to the liver, gallbladder, or pancreas. Okay, there are too many red blood cell dying or breakdown going to the liver is overloaded or damaged. Jaundice is usually symptoms of liver damage. This happens, your damaged liver cell cannot process the bilirubin. Next question is tubular function test includes serum bilirubin, acid load test, HDL cholesterol, both A and B. So tubular function test basically includes acid load test. Okay. So tubular function tests include urine concentration test, urine dilution test, acid load test, and phenol sulfophthalate test. This four comes under tubular function test. Next question is when accurate determination of GFR is required, which clearance test is preferred? Creatinine, urea, inulin, ammonia. Just now I have discussed inulin. Inulin is the polymer or carbohydrate that basically used for the calculation or the measurement of your GFR. This question has several times asked in the exam. Okay, do not confuse with inulin with insulin. Okay, moving to the next question. Inulin clearance test we have just discussed. Next is uric acid test. Why this uric acid test is used? For the renal function test, liver function test, thyroid function test, or lipid profile. Uric acid basically used for the liver function, determination of liver function. So bilirubin uric acid test, this test is based on the response of liver to the benzoic acid administered as sodium benzoate. When the sodium benzoate is either ingested or injected intravenously, the benzoic acid radical is conjugated with aminoacidic acid in the liver to form a uric acid, which is excreted through kidney. Okay, the test related to carbohydrate metabolism for liver function test is serum aspartate transminase, galactose tolerance test, serum alanine transaminase, serum alkaline phosphatase. Your right option is galactose tolerance test. The galactose tolerance test is when galactose converted to glucose exclusively in liver, but the metabolic process is impaired in hepatocellular damage. In this test, person is basically given with 40 grams of galactose dissolved in water If he has normal liver, he should not excrete more than 3 gram of galactose in 5-hour collection of urine after administration of galactose. Okay, this is basically galactose tolerance test. Next, last question of the today's lecture is cholesterol level increases in which condition? Hypothyroidism, uncontrolled diabetic mellitus, nephrotic syndrome, all of these. Remember, all of these options are associated with increase in liver function test. Okay, here the right answer will be option D, all of the above. Okay, so enzymatic method for estimation of cholesterol is cholesterol. Commonly available cholesterol reagent are commonly combined all the enzyme into a single reagent. This reagent typically used for the bacterial cholesterol ester hydrolase. The total concentration is basically increased in case of hypothyroidism, uncontrolled diabetic mellitus, as well as nephrotic syndrome. Okay, this all that's all for today's class. Thank you. We will meet with a new series of MCQ in the next class. Thank you.